second term as president after our recent elections. Our chapter looks forward to providing everyone with quality content this season, as our meetings will stay virtual until further notice. Next month, November 24th, we will have Larry Rosen on our live stream, who will give a presentation about the birds and wildlife of Southern Africa. During his trip, he saw many amazing sights, and it is sure to be a great program. Check out our website and Facebook to stay updated on our upcoming programs, and don't forget to subscribe to our channel. This evening, I would like to welcome Dr. Anna Forsman, a biology professor and the Genomics Lab Research Coordinator at the University of Central Florida. Last spring, I took her ornithology class and afterwards had the pleasure of assisting with her Purple Martin Project as a research assistant. We are all happy to have her as our guest speaker today and look forward to learning more about her research. If anyone has questions, please type them in the chat box and we will answer them after her presentation. Thank you very much for being here today, Anna, and I will pass it over to you.
Okay, great. Thank you so much, Stephanie, and welcome everybody. It's so nice to be with you here this evening. Last year, around this time, I visited in person and talked to you about my master's research with house wrens and extra pair paternity and house wrens. And at that time, I told you that we were ramping up at UCF to start up the UCF Purple Martin Project. And so I am back this fall to tell you all about how that panned out for us, COVID and all, and to let you know some of the preliminary results that we have gotten from our first season and some of the things that we hope to do going forward. Let's see, there we go. So as Stephanie said, I am research faculty at the University of Central Florida. I've been here since 2016. I am an ornithologist by training, and here at UCF, my two main study systems are the Purple Martin and the Florida Scrub Jay. However, I also study a lot of other species, including sea turtles and eastern cottontails and manatees and frogs and reptiles. And the reason for this, my broad taxonomic breadth, is that I have an underlying interest in genomics. And that is the reason why I ended up at UCF. So I run the genomics core facility in the Department of Biology, and I'm also part of the genomics and bioinformatics cluster at UCF. So these are a couple pictures from my lab where I teach the genomics course to undergraduate students and also where I conduct a lot of my research. So at this point, you may be asking yourself, what exactly is genomics? And so genomics is the study of all of the genetic material that you have inside your body, or that any organism has inside their body. So in contrast to genetics, genomics studies all of the genetic material as a sum total, whereas genetics instead focuses on one or two genes at one time and to understand the heritability of those particular genes and the function of those genes. But again, genomics is a much larger scale endeavor. And so how does one do genomics? Well, the basis for genomics is DNA sequencing. And DNA sequencing technology has come a very long way over the last 2 decades. And we now have the ability in even the lab at UCF to sequence entire genomes, or to look at how genomes are expressed by looking at the transcriptome or the transcripts that are produced from those genes, or even to characterize the microbiome or microbial communities inside your gut, for example, or on the surface of your skin, using their genetic or genomic blueprint. So because I use genetic techniques, or the DNA molecule, to understand ecology and biology, behavior, evolution of birds, I consider myself to be a molecular ecologist. And last time when I spoke with you, I was telling you specifically about the house wrens here, using the DNA molecule to characterize paternity to understand mate choice preferences in these house wrens. But there are other ways that you can use DNA. And one of those ways is to, as I mentioned, characterize microbial communities in the environment, or parasites, or other organisms that might be difficult to see with the naked eye or to detect, but they leave traces. They leave DNA traces that allow you to use molecular forensics to figure out who they are and what functions they have in the community. So specifically, I'm interested in how hosts, so birds specifically, interact with those microbial communities and parasites in their environment, and how those organisms influence the health and immune function of those birds. So, but I'm getting a little bit ahead of myself here and telling you about my my broader research question. So I want to back up a little bit and introduce you to the star of the show. And we'll come back to some of these research questions later on in the talk. But the star of the show tonight is the purple martin, of course. And so the purple martin is a swallow, which means that it belongs to the family Hirundinidae. And there are quite a few swallows in the world. There are over 80 species worldwide. And we have about 29 of those species here in the Americas. They have a very broad or cosmopolitan range. So here I'm showing you the range of the barn swallow, for example, a very widely distributed species. And you can see that they reach all over the world. Uh-oh. There we go. Pressing the wrong button here. So the purple martin is a neotropical migratory bird. It spends the summertime, so the northern hemispheric summertime, breeding here in much of North America. And then during fall, they migrate down to South America, there are a lot of birds spending their winters, or our winters, in Brazil and surrounding areas. The purple martin takes the prize for being the largest North American swallow. And the males, the older males, have this brilliant purplish blue iridescent color, and the females and the younger individuals have a bit more of a dusky belly. So purple martins, they have a wingspan of about 40 centimeters. So they have the wingspan that's approximately the same size as a bowling pin, and then their length is about half of that. They weigh anywhere between 45 and 60 grams, depending on the subspecies, which my son likes to remind me is about the same weight as a large kiwi fruit. So purple martins are sexually dimorphic, which means that the males and females look different from each other. So the bird over here, that brilliant bluish purple iridescent color, is a male, an older male individual. This male is at least 2 years old. And this is a female next to him, so you can see that more dusky belly. Still has the bluish purple iridescence here on the back and on top of the head. And so in addition to them being sexually dimorphic, they also have delayed plumage maturation, which I alluded to previously. And so we know that this male here is at least 2 years old, because males do not come into their full adult plumage until their second breeding season. And so during their first breeding season, when they're 1 year old, they look much more similar to females. And so they can be a bit difficult to tell apart from each other at that point. So purple martins are aerial insectivores, which means that they hunt bugs in the air. And interestingly, with purple martins is that they tend to forage at very high altitudes. And so they're flying around and catch lots of different types of bugs. They seem to really love these dragonflies and mayflies, as we'll learn a little bit later. So purple martins lay between 3 and 6 eggs in general, and these eggs are incubated by the females for about 2 weeks. The nest cup consists of, in this case, pine straw or twigs and grasses, and they are often lined with leaves. So here in Florida, we often see a lot of live oak leaves in our nests. Unlike tree swallows that I worked with previously and those house wrens, the purple martins actually have a very, very shallow nest cup. And so it's really more of a platform than anything else. Once the chicks hatch out, they stay in the nest for about a month or so. These birds are altricial, which means that when the chicks hatch out, they are small and pink and naked, and they are unable to thermoregulate on their own. So they can't keep themselves warm on their own, and so parents have to brood. These are pictures that I've taken here at UCF from this last season. You can see the live oak leaves that I was telling you about here. These chicks have just hatched out here at the bottom. These chicks are about a week or so old, and then these chicks here are getting ready to fledge. So you can see that iridescence is starting to come in, even some on the head here on this guy. So there are 3 subspecies of the purple martin. Here on the East Coast, of course, we're most familiar with the eastern purple martin, which we often see congregating around these human-made nesting sites. So we'll come back to the eastern purple martin since that's the species we have here locally. There's also the western purple martin. The subspecies breeds in the Pacific Northwest. And although these birds, of course, also are cavity nesters, we haven't had as much luck getting them to nest in nest boxes as the eastern purple martin, but there are quite a few groups out on the West Coast now that seem to have gotten the hang of it. And so here are some pictures from the Olympic Peninsula Audubon Society, a project that they have out there for putting out nesting structures for the western purple martin. And then finally, probably my favorite, is the desert purple martin. And these birds are just so cool. They are the smallest subspecies, and they live out in the Southwest, and they nest in these saguaro cacti. So natural cavities that have been excavated by woodpeckers, old woodpecker holes. This is where they nest. These guys, like the other subspecies, are colonial, and so you'll often see them nesting in close proximity to each other. These purple martins, we don't know as much about this subspecies as we do about the other 2 subspecies, but there is more work that's starting to happen. And so if this is something that you're interested in learning more about, I really recommend that you check out the Tucson Audubon Society's website. They have lots of really great information. They have just started, over these last couple of years, they have started a Desert Purple Martin project. And they're learning a lot from these birds. And there's some really cool work where they're using endoscopy actually to be able to look inside of those cavities in the cacti. So definitely check that out if you're interested. So all purple martins are colonial, which means that they do like to nest in close proximity to one another. They're cavity nesters. And under historical conditions, they have utilized natural cavities. The pictures that you see here are of eastern purple martins, which are highly colonial and very readily utilize human artificial cavities. So if you are setting up a purple martin colony and want to attract some birds, one of the things you can do is to put out a speaker. And so this is the Songbird Magnet. I just put that picture in there because that's what we've been using at UCF. And it broadcasts the call of the purple martin for a certain number of hours after dawn. And this is a great way of attracting birds to your colony. We also have put out decoys on all of our houses, and that seems to really attract the purple martins as well. So there are a couple tricks that purple martin landlords have up their sleeves for attracting residents. So as I mentioned, historically, purple martins have nested in natural cavities, so cavities excavated by woodpeckers. And this is very much still the case for the desert purple martin here, the cactus, and the western purple martin. However, interestingly, on the East Coast, the eastern purple martins, or I should say across the entire subspecies range for the eastern purple martin, these birds are using almost entirely or almost exclusively artificial housing. And so there are very few places, if any, left where eastern purple martins are breeding in large numbers in natural cavities. Interestingly, one of the last places where they've been documented to use natural cavities is right here in the Orlando area. So at the Orlando Wetlands Park, which I am assuming that probably at least a couple of you have been to go bird watching. And so we are very, very interested to figure out whether these birds are still using natural cavities here, and if so, where they are doing so. If you are someone who likes to go out to the Orlando Wetlands Park and would like to help us out in a new scouting project that we're setting up, I would love for you to contact me. Stephanie, of course, has my contact info, and you can look me up on the UCF biology website to find me there. We'd love to have folks participate, basically to start monitoring the wetlands to see if we can find purple martins using natural cavities there. In 2018 and 2019, the Purple Martin Conservation Association came down and spent a couple days at the Orlando Wetlands Park, and they were not able to locate the birds. Of course, they may have been looking in the wrong place, or they were there at the wrong time, but they did not see any birds. I was there, I guess, at the right time last year, the right place and the right time, and I did see purple martins and purple martin fledglings. So I have high hopes that we'll be able to spot more purple martins out there. So that is one of the projects that we're starting up here in this coming spring. So purple martins have a long history of association with humans, and like us modern culture, I guess, we like to set up bird houses, not just for purple martins, but bluebirds and other species. It turns out that we are not the only ones to do this, and there's quite a long history of doing this and utilizing natural materials such as gourds, hollowed out gourds, to provide housing for birds. And in fact, it's been documented as early as the 1800s that the Chickasaw and the Choctaw peoples were putting up gourds outside their housing to attract purple martins, which I think is amazing. And to this day, a lot of the housing that purple martin landlords utilize are gourd shaped, in fact. So now mostly they're made out of plastic, but many of them still retain that gourd shape, which I think is really neat. So why were Native Americans attracting purple martins? Well, we don't really know, but we can guess, of course. And so the one aspect may be for natural agricultural pest management. So we know they're insectivores, and so they are very efficient at consuming very large quantities of insects. And so it's possible that the Native Americans were thinking about that and therefore wanted to attract these purple martins. I'm very sorry to say, though, that martins don't eat a lot of mosquitoes. We don't have a lot of evidence that they consume a lot of mosquitoes, although that is a misconception that's out there for sure. But again, we're going to talk about this a little bit later on in the talk, and we'll figure out whether that's actually the case or not. But bugs aren't the only type of pests that are out there affecting agricultural fields. And so it's also possible that having these larger colonies of martins could help to chase away other bird species. Of course, this is an oriole that's mobbing a corvid here. But purple martins do the same. I've seen them do that on campus with hawks that are getting too close to their houses. So that's all quite possible, but then again, it could simply be for aesthetics or for cultural reasons. We tend to love birds of all different kinds, and the purple martin is no exception. And every year there are festivals dedicated to the purple martin and the appreciation of purple martin, and it could very well have been the same case for the Native Americans, and they just enjoyed having the birds around. And in fact, this has continued on, and there are thousands, if not tens of thousands of people throughout the United States and Canada that host purple martins in their own yards and serve as purple martin landlords, providing safe housing for them, safe and clean housing, and also contributing to community-based science or citizen science by collecting really important data and submitting that to the Purple Martin Conservation Association, for example. The PMCA has been collecting nest records for the last 25 years from purple martin landlords throughout the continent and have now accumulated over 1 million nest records. So if you're listening to this and you are a purple martin landlord, or you are thinking about becoming a purple martin landlord, please check out the PMCA. They have a great website with lots of great information. And if you are landlording, it would be wonderful for you to contribute those data to the PMCA, to contribute to our understanding of the purple martin, natural history and migration and biology and ecology. So here in Florida, we get to see the purple martins during the late winter, spring, and by early summer, they have really headed off to their wintering grounds. So they breed in much of the United States and up into Canada. And then, as I mentioned earlier, they migrate down to South America for our winter time. We start seeing purple martins here around Christmas time. So late December is when we start seeing those first purple martins. And by early to mid-January, they're pretty widespread here in Florida. We know this because of citizen scientists that are out, folks like you and me, they're out bird watching and contributing those observational data to eBird, for example, through the Cornell Lab of Ornithology, or the Purple Martin Conservation Association. So the PMCA has a scout arrival study that, again, I really suggest that everyone contribute to if you're out watching birds and you see purple martins. So this has been going on for the last 22 years. They've collected over 70,000 observations during that time. In fact, just during this year, they received over 3,000 reports submitted about the first purple martins that are arriving in these different areas, even divided in by age, right? Because we have that delayed plumage maturation. So we can differentiate between adult individuals that are arriving and subadult arrivals. So I just want to focus in here so that we can see the greater Orlando area, and it is looking very, very sparse down in Kissimmee. So I again hope that you guys will get out there. I know you're already out there, but that you contribute those data to the PMCA so that we can learn even more about these awesome birds. So in addition to observational data, we also have tracking data. And so there's a lot of work now that these tracking devices have become miniaturized to the point where we can safely attach them to birds. In fact, some of these devices are so miniaturized now that we can even put them on insects, which I think is absolutely amazing. And so these tracking devices actually let us record what routes these birds are taking and how long it takes for them to get there. So we can learn a lot about migration from these types of data. And we'll come back to this a little bit later too, because I'm hoping that the UCF Purple Martin Project will be able to contribute specifically to these type of data going forward. All right, so now that I've told you a little bit about the natural history of purple martins, I want to switch gears slightly to tell you about my research lab at UCF and also the Purple Martin Project. And I've got lots of pictures to share with you so that you can see our journey to where we are now. So my lab, the Wild Symbioses Lab, consists, of course, of myself and a bunch of really enthusiastic students. So you may recognize some of these people. Of course, you recognize Stephanie over here, who's been a member of my lab. She continues to be a member of my lab. And you'll hear in a bit the work that she's been doing with the purple martins and understanding the diet of purple martins. So Julia here, she is working on looking at bacterial communities, specifically salmonella, and how salmonella may be moved between migratory birds and agricultural farms, and how the relationship between these wild birds and agricultural farms may or may not facilitate the movement of such bacteria. And then we have Lauren Puleo, and she is very interested in migration specifically. And so her undergraduate project has been focusing on using local bird banding data to understand how shifts in migratory activity have been occurring over time and in relation to changes in temperature. And Isabel is a new addition to the lab this semester. She was an intern at the Audubon Center for Birds of Prey in Maitland. And she was working there in the lab over the summer, and she's now joined my lab to work on the scrub jay project. And she's looking at, we have hundreds of blood smear slides that we've collected from the scrub jays to look at a health condition of these birds and also look for evidence of parasites, specifically avian malaria. So Isabel is our cytology expert, and she is working hard in the lab to figure that out. So Christine is a student, this is the student that's been with me the longest. She's now a senior at UCF, and she's not working on birds at all. She's working on sea turtles, but using genomic techniques to understand what these sea turtles are eating. And then Brian, he has graduated from UCF, but he still joins us now and then. And he's involved with Audubon still also. I just heard earlier today that he will be leading a tour at the North Shore Birding Festival here coming up in December. And then Victoria is another non-bird person in my lab. She's actually been studying mealworms and gut microbiota in the mealworm because we know that mealworms are able to digest styrofoam, and she's very interested in how the gut microbiota may be facilitating that. So we've got a great, very enthusiastic group of students that are either working on different questions of bird biology or ecology, immunology, or that students that are using genomic techniques to understand foraging ecology or the microbiome. So a very nice, diverse group of people. But regardless what they've been studying, they've all been involved in the Purple Martin Project, which is super exciting. Before I go on, I also just want to point out that several of these folks have been instrumental in establishing the Nighthawk Audubon chapter at UCF. So of course, Stephanie and Julia were the founding officers, and after they left the president and vice president positions. Then Lauren Puleo took over as president. She is still the president. And then Brian was the vice president until he graduated. So the students in my lab are very active members of this chapter, and I am their faculty sponsor. So it's a really great group of students. So as I said, they were all involved with the establishment of the UCF Purple Martins. I don't think we could have done it without them, actually. And I actually had students from my classes that came out too, just for the fun of it. And happily, this all happened before COVID really moved in. And so you'll see that we're not social distancing. There are no masks in these pictures. And that's because they were taken back in February earlier this year. It's hard to imagine that there was a time where we could do this so recently. But anyhow, so around campus, we were able to put up 12 of these gourd racks with a total of 144 gourds, which is super exciting. We have a lot of ponds around campus, and these birds really do like to nest near water and to forage near water. So we were very lucky that we were able to put up all 12 of these gourd racks around bodies of water. The birds, not only do they forage at high altitudes, they also like for their nests to be situated higher up. And so these poles If you're landlording these poles, you should aim for having them between 12 and 20 foot tall. Of course, we want to make sure that they're equipped with predator guards to prevent snakes and raccoons and other potential predators from climbing up and eating the adults while they are roosting, or the chicks once they're in the nests. And again, they're colonial, so we are putting multiple gourds up on each rack. So during this first season, one of the things we really wanted to figure out was whether the birds had a preference for density of cavities. And so of these 12 gourd racks, they were paired. So we would put 2 of these gourd racks on each pond, of course, situated a little bit apart. And then on one rack, we would put 6 gourds, and on its partner pole, we would put 18. And we're hoping over the next couple of years to record data to see whether the birds have a preference for high density or low density of cavities when selecting their nesting sites. Another thing we did this first season was to look at how the addition of pine straw to these gourds by us at the beginning of the field season influences nest selection or cavity selection by these birds. So as you can see at the bottom here, we have a trash bag full of dry pine needles or pine straw. And so we stuffed every other gourd with pine straw and left the other half of the gourds without pine straw. And as you all know, COVID came in and put a wrench into our field season, in addition to a lot of other things. But one of the things that we were able to figure out from this field season was the fact that the birds really do prefer this pine straw. And so going forward, during our upcoming Purple Martin season, we will continue doing using the pine straw, which is that's a very typical thing to do by citizen scientist Purple Martin landlords. So we will continue with that, and we have overwhelming evidence from just that one season that they do prefer that pine straw. So again, here are some of my students. You see Lauren and Julia here are stuffing those nests, those gourds with pine straw. And here Lauren is trying out the winch system to crank up that gourd rack to the top of the pole so that the gourds are safely at a safe and preferable height for those martins. So as I said, we had a lot of people involved. We managed to get these gourd racks up and all of the gourds, the 144 gourds, in about 2 or 3 days. And that was right before spring break. And so we all dispersed and waited for the martins. And when we came back, the Purple Martins had in fact found us. And so we spotted birds at several of our houses, which we were super excited about. We didn't know whether we were going to be able to attract any birds during our first season. So we were very, very happy to find birds at multiple of the houses. And just to orient you here, hopefully you guys can see my cursor, but this object right here is that little speaker I was talking about, the Songbird Magnet that broadcasts the Purple Martin song. And as you know, we get a lot of rain here in Florida during the spring. And so I actually have just put a shower cap over that speaker, and that seemed to work pretty well. So we had no destroyed speakers during the spring, which was great. These two are actual birds. This third one here is a decoy. Same thing over here. This bird here is a decoy, and the others are real birds. And you can see the back side of that holder for the speaker. So the birds did in fact come, but so did COVID. And as many of you know, UCF shut down at that point and students did not return. So our field season that we were so much looking forward to didn't quite happen the way that we had expected it to. Luckily, though, I live close by here, and so I was able to go out on a semi-regular basis, at least to monitor the houses to figure out where the birds were nesting, when they were laying eggs, how many chicks, how many fledglings. And so I'm very excited to report that we, in fact, ended up having 12 nesting pairs, which is quite a bit more than what I expected. And we produced a total of 50 fledglings. So we're looking forward to this next season. Hopefully these breeding pairs will come back to us and maybe bring their friends, hoping that we'll be able to up that sample size every year. So my students, unfortunately, were not able to be a part of the monitoring this season, but instead I had a very enthusiastic second grader who has, well, he's a second grader now. He was a first grader when these pictures were taken, I guess, in the spring. But of course, elementary schools were closed down as well. And so he came out with me in the field to check on the birds. So it's important to have good staff in the field. So one really exciting part that came out of this, even though the field season didn't turn out the way that we had expected, was that the Purple Martin Association really showed their support for us in many different ways. They very much helped us out in figuring out what housing configurations to go for, and advised us on all manner of aspects of our season. And then they even reprinted an article that UCF put out about my lab and the UCF Purple Martin Project, which was really exciting. So we made the front cover of the spring issue of the Purple Martin update. My students. So lots of fun, even though we weren't able to get out there. I feel like we got a lot of press and a lot of excitement around the UCF Purple Martin. So I think all in all, it was very much a success. So we didn't get very much done science-wise other than attracting the martins this first season, but we certainly have a lot of things lined up going forward. And so some of the things that we're interested in addressing in my lab is, of course, understanding health and disease in birds. I already told you at the beginning of my presentation that I'm really interested in microorganisms and parasites that come into contact with birds and how that influences their health and immune function. So that's definitely an aspect that we're looking at. Also foraging ecology. So I told you earlier that my student Christine has been working on characterizing diet composition in sea turtles using genomic techniques. Well, we've been doing the same thing with purple martins, and that's where Stephanie comes into the picture. That's the project that she's been working on that I want to tell you a little bit more about here in the next couple slides. But in addition to this, of course, we're looking at genetics and genomics of these birds. I'm trying to understand their behavior, specifically migration, and again, foraging ecology. And of course, with my associations with the Purple Martin Conservation Association and Disney Conservation, we are very much interested in the conservation of these birds. And we feel like it's so important to understand all of these other components to be able to address conservation. So let me tell you a little bit more about this diet project that we are working on now with the Purple Martins. So we know that they love to forage on these dragonflies. And we don't have a lot of evidence, as I mentioned earlier, that they eat mosquitoes. But other than that, we don't really know that much about what Purple Martins are eating. So how are we going to figure this out other than capturing the birds and pulling the food out of their mouths and identifying all those insects, which you certainly can do? Well, I can't do that. I'm not an insect expert. But the one thing that I can do, given my background in genomics and using molecular techniques, is to look at fecal samples collected from birds. So that's right, my lab works with bird poop samples a lot. We use these types of samples for several reasons. One, to identify diet, but also to look at these microorganisms that are inhabiting the gut habitat of these birds. What's nice about these fecal samples is that they're also easy and inexpensive to collect. So let me explain what I mean by looking at diet composition based on these fecal samples. So of course, it's not a purple martin, but it is a bird. And this bird then would be eating a mixed collection of insects and other things that would be going through the digestive system and coming out on the other end as a fecal sample. Well, what's great about these fecal samples is that they contain lots of DNA. Not only do they include DNA from the host or the bird that's been picked up along the way through the digestive tract, but there's also lots of great DNA from all of those insects that the bird was eating. And so in fact, we can use these fecal samples and specifically the DNA coming from the insects that the bird's eaten to do fecal forensics. And we can thus backtrack using those DNA sequences compared against a known database of insects to figure out what they're eating. But we can also look at bacterial DNA and parasite DNA to get a better understanding of the health of the bird and parasitism of that bird. So there's lots of great data that we can collect from these fecal samples using these genomic approaches. So once we get those fecal samples, either collected here locally or sent to us by collaborators, we bring them into the lab and spend a lot of time processing those samples under very, very clean conditions so that we don't get any contamination from our own hands or other materials in the lab or cross-contamination across samples. And so those samples are processed and prepared for DNA sequencing. And what I wish I could put here is actually a picture of Stephanie sitting at this hood, because Stephanie's work in my lab has very much consisted of her sitting at this hood processing fecal samples, homogenizing those samples and subsampling them to prepare them for DNA sequencing. And over here, this is a picture of a DNA sequencer, which we are lucky enough to have one now at UCF. So we can actually do this DNA sequencing in-house, which is super exciting. So we started off this project about a year ago. We started talking about it. That's when Stephanie started working with me in the lab. And we wanted to see whether we could actually do this, first of all, whether we could use these DNA metabarcoding techniques to look at purple martin diet and to see if the technique was specific enough that we could detect geographic variation in diet composition across the range of the purple martin. And so since that time, I have been collaborating with Joe Segrist here. So that's the president of the PMCA, the Purple Martin Conservation Association, and Jason Fisher. Jason works with Disney Conservation, and he's in charge of all those wonderful purple martins that are nesting over at Disney. And so the collaboration started there between us three, and then of course Stephanie working in the lab. And then we brought in Kevin Frazier, who is a faculty member up at University of Manitoba. So he's up here, has a lot of purple martins nesting there, and works extensively on these birds. And then more recently, we pulled in a PhD student up in Pittsburgh, Brandon Honig, and he is a specialist when it comes to analyzing these sequence data. So I'm super excited to have all these people on board. We were able to collect samples in 2019 from all three sites. So here in Orlando, up in Erie, Pennsylvania, which is where Joe is based out of, and then Kevin's samples up in Manitoba. And so we have about 150 samples. These samples are being processed right now. Stephanie is the one who did the initial processing of them. And we are getting ready to sequence all of those samples this upcoming weekend. So that is super exciting. And you're probably sitting there thinking, what? You're not going to tell us any results? Well, I have good news for you in that I can't tell you all of the results because I don't have them yet. However, as we were testing out these methods, just to make sure that we could actually use them for purple martins, we did do an initial sequencing run. And so what I'm going to show you next is actually what we found from birds. And this is at all three sites. So this is not just for Orlando. So here I'm showing you some pictures that we were able to identify from those fecal samples that we did sequencing for. So we were surprised to find a lot of moths. So here we see the dark sword grass moth in the top left corner, the gray hawk moth, and the bent line carpet moth. We found a lot of different crane flies, but also beetles, which maybe is a little surprising also thinking about the way that these birds forage. We found evidence of bot flies, lots of different types of midges, again, more of those moths, which suggests that these birds may be foraging at times of day that we were not necessarily expecting that they were. We found evidence of them consuming ants, which is interesting because there have been reports that purple martins may be consuming fire ants. And so that's one of the things that we'll be looking for in these sequencing data as we get them from the larger data set. Again, more midges, some aphids, and of course, those dragonflies and damselflies. This I thought was interesting, this big rice beetle. And again, more crane flies and some other type of beetles here, horn flies and another dragonfly, of course. I think that's my last slide of diet composition pictures. So this is super exciting, I think. This is a very new field in the ornithological literature, using these DNA methods to understand what the birds are eating. And I'm very excited to have Stephanie working with me on this, and look forward to be able to share these data with you as we're producing them at UCF. So if you haven't checked it out already, you should definitely check out my website at UCF. You can learn more about my students and the project that they're working on. And we have lots of pictures from the UCF Purple Martin projects and news as data are unveiled and we are learning things about these wonderful birds. So one of the other things that I wanted to tell you about before I wrap up this evening are some of the other Purple Martin related projects that we're working on at UCF. So earlier we were talking about tagging birds, and that is one of the things that we're hoping to do in the future. And so the MODIS wildlife tracking system is a very large network of these radio telemetry towers that are actually found across the world. And they're used for picking up signals from these small tags that can be attached to birds. And there are some, as I mentioned earlier, they're small enough to be put on insects even. And so these MODIS towers are put together and managed by individuals or organizations. And then the data that are collected from these tags, so not just the tags that you yourself may be putting on the birds, but tags that other individuals have put on tags in other places, if that bird is going by your station, all of those data are being picked up and delivered to a central data repository that is available to the public so that everyone can share in that wonderful wealth of data. And so we are hoping to be able to establish one of these towers, one of these MODIS towers at UCF, and to begin tagging our own birds here so that we can follow them on their migrational journey. So maybe in 2021, because of COVID, it may be delayed by a year or so, but hopefully this will be coming soon. So this MODIS wildlife tracking system, if this is something you're interested in, again, I recommend checking out their website because there's lots of great information and maps available there. So there are in fact almost 1,000 of these MODIS towers that are spread out across 4 continents. We've been able to use these MODIS towers to track over 200 species, not just birds, but again, insects, bats, so lots of great information coming from here. And in fact, almost 25,000 different or 25,000 individuals have been tagged and then followed with these MODIS towers. So what makes these MODIS towers so powerful is when they're arrayed in a way that you can you can pick up individuals that are migrating or moving through these areas without missing anybody. So for example, here along the coast between Charleston and Savannah and further south, we have this really nice array here. So birds that are coming down and moving down the coast will be able to be picked up along that path. Another great array here is across the state in Florida. And so there are definitely some gaps here. We'd like to have some more towers here so we get a nice coverage across the state. But again, birds that are coming through and migrating through Florida will hopefully be picked up by at least one of these towers along the way. Now, oops. As you can see, it is very empty here in the Orlando area. And so we're hoping to change that sometime soon. So that's our upcoming MODIS program. The other really fun thing that we've been doing at UCF is partnering with the student interns at the UCF Arboretum. And as I mentioned earlier, historically, folks have been using natural gourds to attract purple martins and for them to nest in. And so we partnered with the students to grow these gourds. So the students have been working hard out in the field doing the research that they needed to figure out exactly what type of structure for these gourds to grow on. And we're super excited when it's ready for harvest. We were going to dry them out and make them into houses. And we won't be using these houses for the UCF Purple Martin Project. This is more, I would say, of a fun, inspirational, and just team building activity, especially during these COVID times. And hopefully the students will be able to use this to maybe do some fundraising too, to support their activities at UCF. So I think just a great opportunity to get students involved, both in the field working with the birds and in the lab working with samples like Stephanie, and even these Arboretum students working with these Purple Martin gourds. So that's the UCF Purple Martin Project in a nutshell, where we are at this point. Again, super excited about what the next season has to offer. And I look forward to coming back and telling you about the more results on the diet study and other upcoming studies as our sample size increases over the years at UCF. So with that, I am happy to take any questions about anything I've talked about today or anything else related to Purple Martins or my lab or UCF. So thank you so much for having me.
Thank you so much. What a great program and lots of good information. And I do have some questions for you in the chat and then a couple of my own, if you don't mind answering them.
Sure.
So I have one question. Yes. One question is there any way to ascertain how the massive wildfires in the Amazon this year are either affecting migrating purple martins or are they likely to?
That's a great question. So I am working with a team of purple martin experts, and some of them are located in Brazil. So I work with the International Purple Martin Working Group. And we meet every week to talk about the research that we're doing, upcoming projects and proposals. And we have this international exchange of data and well, we'll see now with COVID, but otherwise folks from up north have been going down to Brazil during the wintering months to catch birds, put trackers on birds, collect morphometric and health measurements, screen for diseases. So we're actively working with folks down there. We actively have projects going. And so this is very much a topic of conversation right now during our meetings about what things are looking like down there and how we can assess the effects on the purple martins. I can tell you that even during this talk, this 1 hour talk, I have 4 notifications on my phone from folks in the Purple Martin group that are talking specifically about what's going on down in Brazil. So it is very much in the works, and people are talking about this and and we we hope to be able to provide some answers, but I don't have any definitive answers to give you right now, unfortunately.
All right. Thank you for answering that. And it actually answers the next question about were there any organizations in South America collaborating with those of you studying this lovely and unique swallow in the USA? So that's great to hear that you are in constant contact with that group.
Yeah, it's a very vibrant group of wonderful people involved. I'm very, very excited. I'm a newcomer to the group. I've only been part of the group for about the last 6 months, but lots of great ideas flying around there, and I'm very excited to see where that leads.
All right. I have another question. Does the Purple Martin Project involve banding at all?
It will. It has not involved any banding at this point. Everything was put on hold because of COVID, but yes, it will absolutely involve banding.
Okay. And now I have a few more. So, has there been any house sparrows or any other species attempting to nest in the gourds?
Great question. Right. So house sparrows are known for being terrible and using any cavities that they can get a hold of. And this was definitely a problem for me when I was working on my PhD work up in Ithaca, New York with tree swallows. We always had those silly house sparrows that were coming in, and they would kill adults and kill chicks, and they would break eggs. And so I'm very happy to report that we haven't had any house sparrows find our gourds yet. I don't typically hear house sparrows on campus, so I'm hoping that we'll be able to keep them at bay for a while longer. Of course, the other culprit are starlings, European starlings, and I did hear them. I heard a couple starlings close to one of the nesting sites early in back in February, I guess it was. But they never did anything to our nest, and I think one of the reasons for that is the houses that we purchased are supposedly starling proof, and so the opening to them is supposed to prevent the starlings from coming in. But we just don't have a lot of starlings.
Okay. Another question is, are the purple martins able to eat venomous caterpillars?
Oh, I don't know. I don't know that. But we can maybe figure that out by looking at our sequencing data. The caterpillars, yeah, I don't know. I don't know enough about these insects, but that's a great question.
Definitely. Okay, here's another one. You mentioned on campus you have the 12 gourd racks with 144 gourds. How many more gourds can each rack fit?
Great question. So the racks will be able to accommodate 24 gourds each. So there's definitely room for expansion. And so we have taken up the best sites on campus at this point that we have access to, because these birds really want open areas, preferably near water. So they're very picky about how close their houses are to any sort of trees or even shrubs. So if our numbers continue to increase over the years, we will likely expand by adding more gourds to the houses we already have, as opposed to adding more houses. There is always the possibility of adding more houses, but as I said, we've already chosen the most select sites. So I think upping the gourd numbers is the way to go.
Great. Another one is, do the decoys generally attract many species of bird, or is there a specific one to attract purple martins?
Yeah, there's the ones that we use are black, and that they have a little bit of a white eye spot, and they're these plastic birds the same size of purple martin, and that we put on top of the houses. Um, it's kind of humorous. We laugh now as we drive by or walk by these houses. They don't We probably just didn't attach them tight enough, but almost all of them by the end of the season had flipped around and were hanging upside down. So I don't think they were working very well as decoys by the end of the season, but we don't need them as decoys by the end of the season. So, but they seem to work in conjunction with the speakers to attract the birds.
And will you need to continue using the decoy and speakers next season, or now you could take that away?
I don't know if we need to, but because it's just our second season, I don't think we want to take any chances. So our plan is to put out the decoys. Well, the decoys are actually still out there, but to put out the speakers again this year. Since the colony is still so new, and we have several houses that didn't have any activity at all, so we're hoping to attract birds to those houses as well.
Great. And I have another one. We have a lot of storms. What happens to gourds in very high winds?
Right. So we were just talking about this in lab meeting earlier today. When we first started this, I had anticipated that we would be taking down the gourds at the end of the season and storing them and then putting them out again. But actually, our gourds are still out there. And I consulted with Jason over at Disney Conservation, and they keep their gourds out all year and only take them down when there are storms approaching. And so we are going to take the same approach. Otherwise, I think a lot of landlords that live further north do take down their gourds, clean them out, and store them for the winter. But here in Florida, we're not planning on doing that again unless there's a hurricane coming our way. We will, however, as soon as the temperature calms down a little bit, we're going to get out there and pull those gourds out, take out the old nesting material, clean them out, soak them in 10% bleach to make sure that they're nice and clean, hose them off, and put them back up again so that they're nice and clean and disinfected for the birds coming back this next season.
Excellent. Perfect. So I have another question for you. Will you be looking at the health of the purple martins with the fecal samples at a later time? For example, looking for parasite or bacteria in the data?
Yeah, so the gut microbiome we are already looking at. And so that sequencing run that I'm starting up this coming weekend, we're looking at diet using two different targets, two different genomic targets that may tell us different things. And then we're also using a genomic target that will allow us to differentiate between different microbial taxa. And so, yes, we are already doing that for those samples that were collected in 2019 for those three different sites, which is super exciting because we'll be able to look then at geographic variation in gut microbiome composition and also how that microbial composition relates to the diet of those birds. So yes, that's definitely happening. I have plenty of DNA left over from those analyses. And so looking at parasites, worms, for example, is something that we can go back and do later on. We would need a most likely a different genomic target to be able to look at that. But as I said, we have plenty of DNA left over, so we certainly can do that going forward.
Excellent. And then I have a final question for you. Will you be analyzing new fecal samples each year to add to kind of like a year by year picture of their diet eventually?
Yeah, so we're still trying to figure out exactly how we want to go about doing this and exactly what questions we're most interested in asking. I would say looking at year to year variation is maybe for me less interesting than looking at, say, variation across the breeding range or into the wintering ground. So that's one thing that we would love to do, especially since I have collaborators, part of the Purple Martin, the International Purple Martin Working Group, that go down to Brazil and catch the birds and put loggers on them, that there's an opportunity for collecting fecal samples there to look at what the microbial composition looks like on the wintering grounds, because we've learned even from the human gut microbiome that there's quite a bit of overturn. And so you change your diet and the microbiome will change. If you're sick or take antibiotics, the microbiome will change. And so it's very likely that the gut microbiome of these birds is quite different on the wintering grounds, but we don't know that. And so I would say, again, we will continue sampling going forward, but not necessarily for the purpose of looking at interannual variation so much as being able to sample different areas or different life history stages. So for example, comparing adults and nestlings, I'm very much interested in understanding how a diet differs between the adult feeding itself for nourishment as opposed to what an adult feeds the chicks. And hopefully we'll be able to use this diet metabarcoding approach to get at some of those questions. So again, the answer, I guess, the loosey-goosey answer is that we don't really know. We're still developing a lot of these questions, which is part of the excitement, I guess, of having a new project like this.
That's great. And just so everybody knows, in the description, I did put a link to your lab. So anyone could click the link and check out your lab news and what you're doing and keeping up to date on everything.
That sounds wonderful.
Thank you so much for coming today.
I just want to add too that if there's anyone out there who's interested in landlording, or if you already are a Purple Martin landlord, I would love for you to contact me because that this is one of the plans going forward too, is to kind of connect Purple Martin landlords in the greater Orlando area so that we can learn more about these birds by being able to collect data from a larger group of individuals. So please do contact me if you are interested in participating.
Thank you so much for such a great programming and letting everybody know what your research is and new things happening. And I think it's very interesting also how the Arboretum is growing those gourds, and whether it will attract birds in people's yards, but it's still a fun project to for the students to take part of. And I wanted to thank everybody else for coming out today, spending your evening with us. Every month, like I said earlier, we will be having the live streams. And I will leave any final thoughts with Dr. Forsman, if you had any final comments or anything.
Stay safe, take care of each other, and go bird watching.
Thank you. Have a good evening, everyone, and thank you very much.